This is for question 6A on the take-home exam. The process of cell division begins first with mitosis, which is the division of the nucleus, and then is followed by cytokinesis, which is the division of cytoplasm that results in two identical daughter cells. Mitosis begins with the condensing of chromatin, generating chromosomes that are individually discernible under the microscope. Because DNA replication has already taken place, each chromosome actually consists of two copies that remain attached to each other until the cell divides. As long as they remain attached, they are referred to as sister chromatids. And as the chromatids become visible, the nuclear envelope breaks into fragments, and then in a process guided by the microtubules of the mitotic spindle, the sister chromatids separate and move to opposite ends of the cell as full-fledged chromosomes. And a cell spends most of its time in interphase, which is actually precursor to all of the events of mitosis. During interphase, the amount of nuclear DNA doubles, and the new nuclear DNA is synthesized. And so here I have a demonstration of interphase. And so you can see there's the gap phases here in green, which are the time in between DNA synthesis and mitosis. And so the DNA in this step is copied in preparation for cell division, resulting in two identical full sets of chromosomes. So this is for a 2n=4 diploid cell. And so here we have prophase, where the chromosomes condense into X-shaped structures, as you can see, and that can easily be seen under a microscope. And each chromosome is composed of two sister chromatids, and they contain identical genetic information. The chromosomes pair up so that both copies of chromosome 1 are together and both copies of chromosome 2 are together, and so on. At the end of prophase, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. And then the mitotic spindle, consisting of mitotic tubules, extends across the cell between the centrioles and move to opposite poles of the cell. So here we are in prometaphase, where you can see the microtubule spindles, or mitotic spindles, heading towards the center of the cell. And you see the sister chromatids, here's mitotic spindle, the centromere, which is the center of the chromosome. And so then we have metaphase, and metaphase is where the chromosomes line up neatly along the center of the cell, and the centrioles are now at opposite poles of the cell with the mitotic spindle fibers extending from them, and the mitotic spindle fibers attached to the sister chromatids. Then anaphase is when the sister chromatids are pulled apart by the mitotic spindle, which pulls one chromatid to one pole and the other to the other opposite pole. So then we have telophase. So at each end of the pole, each pole of the cell gets a full set of chromosomes to gather together. A membrane forms, so you can see the new membrane forming around each set of chromosome to create two nuclei, and then the single cell pinches off in the middle, so here's the pinching off, to form two separate daughter cells, each containing a full set of chromosomes within a nucleus. And this is when cytokinesis occurs, and you get the two identical daughter cells. And so as you can see, we begin with the 2n=4, and we end with 2n identical daughter cells.